Okay, well, thank you for the invitation to speak here. My name is Eran Mukamel, and I'm from the Department of Cognitive Science at University of California, San Diego. And I'm excited to tell you about our work as part of the BICCN that you just heard about, integrating single-cell epigenomics data into a multimodal brain cell atlas within the BICCN. And so since a number of the other speakers in this session are going to talk about especially transcriptomic data and cross-species comparisons, I've really tried to focus here on the epigenomic aspects of this broad collaboration. So just to acknowledge some of the folks doing the work, these are some members of my lab, in particular Fengming Shi, Junhao Li, and Ethan Armand, who are involved in this work. And everything I'll show you is based on a really broad collaboration with Honghui Zheng, Joe Ecker, Margarita Behrens, Ed Calloway, Bing Ren, and other members of the BICCN, who you'll hear from here as well. Okay, so our brains are extremely diverse. So for example, here's a picture of two Mukamel Lab interns, my two kids at the ages of 2 and 4. We know that siblings share a common genetic heritage that shapes their brain development and cognitive function, but at the same time, our diversity and the differences between us are really essential to our humanity. And this is true also at the level of individual cells. So if I show you a picture here from one of the founders of neuroscience, Santiago Ramón y Cajal, from 100 years ago, you can already appreciate the extreme diversity of morphologies of brain cells. We know that individual neurons share the same genetic sequence, and yet they have differences in their morphology, connectivity, physiology, and gene expression. And one of the focuses of my lab is really trying to understand how does DNA methylation support the brain's development and plasticity and diversity in support of cognitive function, which really requires this diverse repertoire of parts. And this is not just a goal for my lab, it's one of the central goals of the Brain Initiative. In fact, the first goal of the Brain Initiative was to define the landscape of cell types in the brain as a starting point for further progress. And I really want to contrast our understanding of biological neural networks of brains with the picture that many of us have in terms of artificial neural networks, which really have achieved remarkable progress and capabilities in terms of mimicking some cognitive functions, but they're built from largely uniform components. So if you think of the neurons in an artificial neural network, they don't have anything like the diversity of the biological neural networks that we know. So I'm going to be talking about how the epigenome contributes to diversity in the brain. And just to remind you, the epigenome comprises chemical covalent modifications of DNA and of the histone proteins that make up chromatin. And one analogy I like to give is that the epigenetic marks are kind of punctuation marks. So they don't change the sequence of letters of the genome, but they can dramatically change the meaning. So you can think of a sentence in English like, some people find inspiration in cooking their families and their dogs. Add a couple of punctuation marks, some commas. Others find inspiration in cooking their families and their dogs. So similarly, DNA methylation marks you can think of as commas. In this case, they change the patterns of gene expression across cell types. And I'm really inspired by this picture from Conrad Waddington, from many years ago, the idea that there's a landscape of cell types in the body, including in the brain, and that the epigenetic programs through development in the adult act to stabilize the regulatory networks that define those cell types. And so with John Nye, a year or so ago, we wrote a perspective arguing that we can think of cell types as a self-stabilizing program within the genome. And then to really understand that, we need large-scale data sets which can allow us to understand the mechanisms for that self-stabilization. So again, I just want to give a little bit of background about what's known specifically about DNA methylation to give you some context for thinking about how these data could be useful for understanding brain cell types. And so we know DNA methylation is again a covalent modification of cytosine in the genome. We know the enzymes, the DNA methyltransferases that catalyze the addition and also the removal of DNA methylation via the TET family of enzymes. And for the most part, I'll get back to this point, but for the most part, we know that DNA methylation occurs at CG sites, or sometimes called CPG sites in the genome. And so there are very well established roles of DNA methylation in processes such as imprinting, exon activation, transcriptional repression. It's often misregulated in cancer. We know that it's a stable and heritable epigenetic mark, so that through cell division, the pattern of DNA methylation is copied. And yet it's reversal and potentially activity dependent. So there's evidence that in neurons, the precise pattern of DNA methylation can be affected and changed by experience and patterns of neuronal activity. And an example of the relevance to cognitive function is Rett syndrome, where we know that mutations in MECP2, methyl cytosine binding protein, cause the autism-like phenotype. And of course, DNA methylation is intimately connected with histone modifications, chromatin structure, other epigenetic and non-epigenetic gene regulatory processes. And as I mentioned, non-CG sites, so cytosines followed by something other than a G, are generally unmethylated. And I'll get back to this point because it turns out to not be true in neurons. Okay, and again, just a very high level overview. What do we know about the roles of DNA methylation? Broadly, well, it can have dramatic roles, for example, in governing flower shape in these epimutants of, again, the same phenotype in these toad flax epimutants, same genotype, different phenotype that's, again, stable and heritable here transgenerationally due to DNA methylation pattern. Similarly, DNA methylation is thought to govern the differentiation between different castes in social insects. There are examples where we understand, such as the agouti gene in mammals, where we understand that DNA methylation is responsible for repression of a gene that controls coat color and also obesity. And again, in the long term, we're interested in understanding how relevant is this for us as human beings. Okay, so a number of years ago, working with the Ecker lab at the Salk Institute and Margarita Behrens, who pioneered the technique of whole genome bisulfite sequencing to measure DNA methylation with base resolution throughout the genome, we discovered that there's really a unique feature of DNA methylation in the brain. In particular, we found that whereas, as I mentioned, the vast majority of tissues throughout the body have almost no detectable non-CG methylation. So more than 99% of the DNA methylation is in the CG context. The brain is really an outlier. So neurons in particular have over half of their DNA methylation, their methyl cytosines are actually at non-CG positions. A distant runner-up are glial cells in the brain that also have some non-CG methylation, and all other stem cells, with the possible exception of embryonic stem cells, all differentiated cells, I should say, have almost no detectable non-CG methylation. And so one of so this turns out to be very useful for our project in terms of analyzing cell types in the brain, and it's also a biological question what the role of this non-CG DNA methylation is. It turns out that this DNA methylation accumulates throughout development, and it accumulates in a rather protracted fashion. So in humans, we're born with none of this non-CG methylation, even though our neurons are already present in the brain and post-mitotic. The DNA methylation accumulates postnatally throughout the first few years of life. In humans and in mice, you have a similar but sped up progression that proceeds over the first few weeks of life in the mouse. And it's also very conserved between individuals. When we look across different individuals, we see that the pattern of DNA methylation, here I'm showing you both CG and non-CG positions, is very, very strongly conserved. In fact, we found that the correlation between two individuals was almost as strong as it could be given the sampling statistics determined by the read depth of our whole genome bisulfite sequencing. Okay. Now, it turns out that the DNA methylation pattern, both the CG and non-CG methylation, are not only conserved across individuals and very unique in the brain, they also have a very cell type specific distribution. And one of the things I really want to impress you with here is how dramatically cell type specific these marks can be. So for example, here I'm showing you the GABA transporter gene that's expressed in GABAergic neurons. So for example, in PV and VIP cells here that have been purified, you see the RNA-seq signature of this gene in the GABAergic neurons, but not in the glutamatergic neurons. And when we look at the DNA methylation pattern, you can see very low levels of DNA methylation, CG DNA methylation at the promoter region and also the first intron, so really the first half of the gene in this case. And in addition, there's a very strong signature throughout the gene body in terms of depletion of non-CG methylation. So these correlations in the gene body between low DNA methylation and high gene expression allow us to link transcriptomic and epigenomic data. Here are some additional examples where you can see genes that are expressed in only one cell type, again, have the corresponding methylation pattern of demethylation in that cell type only. And so this is, again, data that was obtained by purifying neurons with a technique called intact. And as I'll show you, we with single cell data have been able to go one step beyond this. But before, I want to use these data to illustrate the cell type specificity in a slightly simpler context. So sticking here with the intact data for a moment, I can show you the pattern across all the genes in the genome. So here what we did was a simple cluster analysis where we clustered the genes according to their DNA methylation pattern across these 3 specific cell types, as well as some less specific samples. So for example, we have whole adult cortex as well as glial cells. And without going into too many details, what you can see is that roughly half of the genes have a relatively non-specific, non-cell type specific pattern. So you see genes that have a very low methylation pattern across all cell types or a very high methylation level across all cell types. So these might be housekeeping genes. Some genes that are pan-neuronal, where the pattern of methylation is shared across all neuronal cell types. But then what's really striking is you have a third to a half of all the genes that have a very striking cell type specific DNA methylation pattern. So just to give some examples, here the gene VIP is in this first cluster of genes that has a low level of DNA methylation in the VIP cells, not surprisingly, given that this is a marker of those cells. Parvalbumin is demethylated in the PV cells, and VGlut1 is demethylated in the excitatory neurons. And really what we find is that this pattern of DNA methylation is just as cell type specific, if not more cell type specific than the transcriptomic signatures that we also find in these cell types. This intact technique also allowed us to look at attack sequencing, so to probe the open chromatin of these cells. And what we found was very corresponding patterns of open chromatin. For example, here is an example of a region of open chromatin across all 3 neuron types that also corresponds to low DNA methylation across all 3 neuron types. But in addition, there were very cell type specific regions with low DNA methylation and open chromatin just in one cell type. Okay, so the types of questions we're interested in addressing with these data are, can we understand the relationship between epigenetic regulation and gene expression? So for example, can we correlate the level of DNA methylation in bins around the transcription start site with the expression of the corresponding gene? And so when we do that analysis and look at different epigenetic features, so for example, non-CG methylation in blue, CG methylation here in green, or attack-seq in red, what we find is that actually the non-CG methylation is very strongly correlated with gene expression. Negatively correlated, I should say. So it's the correlation of the negative non-CG methylation. And we can build models where we regress the gene expression against a set of these features using lasso to sparsify the regression and find out which features are most predictive and informative for gene expression. Okay, but all of these data that I've been showing you have really been using bulk. Sorry, I just want to check the time on my recording here. Okay, so they've been using bulk DNA methylation and in the sense of purifying populations of excitatory PV or VIP cells. In order to really understand the fundamental limits of cell type specificity in these cells, we need to go to the single cell level. And so starting a few years ago, and you know, really following in the footsteps of the enormous progress that was made in terms of single cell transcriptomics, single cell RNA sequencing, Joe Ecker's lab with Chungwan Luo, who was in his lab and now has his own lab at UCLA, developed single nucleus methyl cytosine sequencing. So this is a technique that can, with fairly high throughput, profile the DNA methylation pattern throughout the genome in thousands of cells. It's more expensive, relatively speaking, than RNA sequencing, so we typically profile fewer cells, but the amount of information that we obtain per cell is really large, since we are able to capture 5 to 10% of the whole genome in individual cells. So to give you a sense of what we can do with these data, here's a TSNE embedding showing several thousand cells from the human frontal cortex. And you know, this is based on DNA methylation features. To annotate these cells, we can take groups of cells, for example, that have formed a cluster, and pile together all of the information we have from all of these cells to obtain a really high coverage, deeply sequenced pseudo bulk DNA methylome for these cells. And using that information across each of the clusters, we can then go ahead and annotate the cell types. For example, here we're looking at the gene LHX6. And just to explain this browser view, what I'm showing here, each track is one of these pseudo bulk samples coming from a cluster of individual cells. The individual ticks are cytosine positions, CG positions, and the height of each tick corresponds to the level of DNA methylation. So what you can appreciate is the level of DNA methylation is very low in the gene body of LHX6 within these 4 clusters arranged at the top of this browser. And these correspond to the PV and SST cells, which are MGE-derived GABAergic neurons that express LHX6. And by looking at combinations of these genes, we can go ahead and annotate the cell types. Okay, so coming to the BICCN now, we collaborated with over 83 co-authors to really try to take a very deep dive into the mouse motor cortex and try to understand, using multimodal data, I think 6 or 7 different RNA-seq data sets, open chromatin using single nucleus ataxic, as well as DNA methylation data. What is the multimodal definition or landscape of cell types of the motor cortex? So here's an overview of the data sets. You can see that the data sets differed by more than an order of magnitude in terms of the number of cells that we could obtain. So with 10x, for example, RNA sequencing, we can very cheaply profile, or I should say relatively cheaply profile many tens of thousands of cells. But the amount of information in terms of the number of unique sequenced fragments that we record for each cell is relatively lower, tens of thousands. Whereas with DNA methylation and also full-length transcriptome sequencing with SmartSeq, we get more information per cell, but fewer cells. With each of the data sets on its own, we're able to cluster the cells and really identify dozens of different neuronal and non-neuronal cell types. So you can see here in each of the analyses, we see excitatory neurons, inhibitory neurons in the reddish colors, and non-neuronal cells. And then we can also use techniques for integrated analysis to bring these together. So this was one of the focuses of our efforts in the joint analysis working group, was to really find ways of integrating the data. So here what I'm showing you is how we view this as a missing data problem, where we treat each data set as having a set of features that we've observed, but also other features that we haven't observed. So for example, cells measured by single cell RNA sequencing, we observe their gene expression, we haven't observed their DNA methylation, and so that information needs to be imputed. And to do that, we take advantage of the strong correlation of the gene body signatures. So if we look at the average DNA methylation, for example, in the gene body of this gene TAC1, it is very low in the parvalbumin expressing cells. Correspondingly, we have a high level of chromatin accessibility within the gene body and also a high level of gene expression. So this gives us a correlation that we can use to link individual cells between data sets and then perform integrated analysis. Now, of course, our goal is not only to look at gene bodies. In particular, we're very interested in distal regulatory regions. For example, here I'm showing an enhancer that's upstream, about 20,000 base pairs upstream of the promoter of TAC1. And you can see low levels of DNA methylation and a peak of open chromatin that tell us that this is a putative enhancer active in this cell type. And so this information wasn't used for the integrated analysis, but we do use it once we have performed the integration to interpret the regulatory networks in these cells. So the technique that we've used to integrate the data is called single cell fusion. This is available from our lab's GitHub. And it's based on the idea of building a mutual nearest neighbors network. And so for each data set, for each cell in each data set, we find the best matching, most correlated cells in the other modalities and use these as links to infer the missing information about the unobserved data. And this really works very well to robustly link data sets. Here you can see an embedding of cells from across each of the eight or nine multimodal data sets that integrate in terms of the cells coming from different data sets, but separate nicely by cell type. To give you a sense of the resource that's available from the result of this, here is a browser view that shows base resolution DNA methylation at the top here, single nucleus ATAC-seq, and it also has RNA-seq tracks available. We're showing with these black ticks here the predicted gene regulatory elements called using a software called Reptile from the Ecker lab. And this really enables us to zoom in on, for example, again, that gene TAC1 that I showed you earlier. You can explore the data in this region across multiple cell types to understand the landscape of regulation and gene expression. One of our interests with these data sets has been to cross-validate the clustering to try to determine where we are on the lumper splitter axis in terms of whether we're splitting our cells at an appropriate resolution. And we developed techniques for doing this by splitting the features as well as the cells that are being used for clustering. I won't dwell on the details here, but just to say that we can do this in a multimodal way by cross-validating across data sets and use that to obtain some estimates of how many clusters can really be reliably reproduced across data sets. And I believe Megan Crow is going to be talking later in the session about techniques using MetaNeighbor for also looking more statistically at the replicability of these clusters. So finally, I wanted to say just a few words about what is the payoff? What can we use the epigenomic data in particular to do that goes beyond the transcriptomic information that we get from these single cell data sets? And one of the things we're very interested in is linking enhancers with the genes that they regulate. So by looking across cell types, we can try to look for enhancers whose methylation level, for example, is tightly correlated with gene expression across cell types, and separate these from other enhancers that are not linked. Here's an example of a nice region within the gene SLCA1A2, where you have a very strong correlation between the DNA methylation signature of this enhancer and the gene expression. We can do this throughout the genome and find examples of enhancers and genes that are correlated. And the correlation is then validated and verified across CG and non-CG methylation, as well as ATAC-seq data that, again, wasn't used for identifying these enhancers. We can then go beyond just identifying the enhancers to try to interpret them. So one really important analysis for us is to look at the DNA sequence motifs that are enriched within these enhancers. And so by looking at transcription factor binding sites in the enhancers, we can interpret which transcription factors may be important for regulating in particular cell types. In this case, this was from our paper comparing human and mouse homologous cell types. We could find examples of transcription factors that are used in a conserved way across the two species, as well as more divergent patterns of transcription factor usage. Finally, Hanqing Liu in the Ecker lab has been using these data to really try to finally dissect which transcription factors are involved at particular levels of the cell type hierarchy or lineage. So here's an example where at the very highest level of this hierarchy, he can find transcription factors that are enriched at one side of the branch point versus another. And then going down further to progressively finer and finer levels of this cell type hierarchy, he can identify the transcription factors that may be responsible for regulating each branch point along this lineage. So just to summarize, I've tried to emphasize how bringing together multimodal data, in particular transcriptomic and epigenomic data, provides a foundation for functional understanding of brain circuits. Non-CG DNA methylation is not something that's typically considered in other tissues, but in the case of the brain, it's a very unique and conserved feature of neuronal epigenomes. Again, it develops postnatally, and it really provides very reliable and robust signatures of neuronal cell type. And finally, we can use these data to reveal the regulatory networks, including cell type specific enhancers. And so I'm looking forward to taking your questions. Thank you.